Thank you very much. First, I would like to thank the Fame Lab to invite me to present the work I've been done. This is part of my PhD project, so I will present some of the results that we have so far. So I will start by talking a little bit about bone tissue. Bone tissue is a living tissue. It's constantly being renewed in a process called bone remodeling. So very briefly, what happens, we have two major bone cells, which is the osteoclasts and the osteoblasts. What happens is that the osteoclasts will start resorption bone, destroying bone, and after 3 weeks, the osteoblasts will start to work and will be responsible for form new bone. So, and then after more or less four months since the beginning of the process, some osteoblasts go into the bone matrix and they will become another very important cells, which is osteocytes. So what happens in bone tissue is that we have this bone remodeling and we have a process of bone resorption and bone formation. So it's very important to keep a balance on this process, because if that does not happen, what we will have is like bone metabolic diseases like osteoporosis. So osteoporosis will happen when there is an imbalance in these two processes, and when bone resorption will go over bone formation, we will have osteoporosis. And osteoporosis, as you can see here, is characterized by low bone mineral density, because osteoblasts are not acting very well. I mean, resorption is over formation. And what happens is that if you have a weak bone with osteoporosis, then we will have a fracture. It will be a matter of time to have a fracture, and that because if you have a fracture, because your bone, your formation, your osteoblasts are not acting very well, what will happen is it will be very hard to heal that fracture, and of course that will impair people's life. It will have an impact on their quality of life. And when we have cases of osteoporosis, it all goes mainly on lumbar spine, femoral neck and forearm. This is the most common sites for osteoporosis. So this process of bone remodeling, it's not the same, it doesn't happen in the same rate throughout our life. For instance, during the growing years, bone formation is very active and goes over resorption. That's why during the growing years, and particularly during puberty, there is a lot of bone mineral density, because the bone cells are responding to some hormones particularly. But then when after a certain age you reach a peak, and then bone formation starts to decrease, so it means that resorption is going over bone formation, and that happens mainly in females and at the menopause, particularly because the estrogen levels decrease and drop significantly. So the process of remodeling changed throughout our life. And what happened, this is the stages of osteoporosis. Usually we start with normal bone, let's say during the growing years, and then if you have some issues you can have osteopenia, which is characterized by low bone mineral density, and then you have osteoporosis, and a fracture will be a matter of time to happen. So it is estimated that 1 in 2 women will suffer osteoporosis and 1 in 4 men. In the US, in 2002, 43 million people have low bone mineral density or osteoporosis, and this prevalence has been increasing, and is estimated that in 2020 around 61 will have osteoporosis. So because it's increasing, it's important for us to understand what might cause osteoporosis and low bone mineral density. So age itself, it's the major risk factors, because if you remember the graph, it will drop the bone formation after a certain age. So this is the major risk factor, and sex as well. If women are at increased risk because of menopause, again because of the estrogens. And then you have genetics. Genetics accounts for around, family study says that genetics accounts for around 60 to 80%. So it has a significant role as well. Nutrition, we all know that calcium intake, energy intake is very important. It's related to bone health. Vitamin D that works through PTH as well. It's very important to have optimal levels of vitamin D in your bone. And growth and the circulating levels of hormones, it's important because if you remember the graph, the bone mineral density will increase during the growing years because there is an increase in circulation levels of estrogens, and that will make the growth hormone increase and the EGF-1, and so it's important to have a normal growth path. If that doesn't happen, the chances of developing low bone mineral density increases as well. And then we have exercise. Physical inactivity, I mean, not doing exercise is related to osteoporosis. So inactivity is also an important risk factor. And because it is one of the major risk factors and major cause of osteoporosis, research has been focusing on the effects of exercise to prevent the disease. So, and they study the effects of physical exercise mainly in two groups. One in non-exercising participants, and then in participants that goes on organized and frequently physical activities like athletes. So what happened is that studies using in their methodology non-exercising participants, they do usually a randomized controlled trial of short interventions of weight-bearing exercise, and the outcomes of this study are very straightforward. They said that if the effects of exercise, it's beneficial for bone. I mean, if you're going, particularly in weight-bearing exercise like plyometric exercise, muscle strength as well, you will improve your bone mass because you are stimulating the osteoblasts. But on the other hand, if we look at athletes, I mean, that goes in a systematic exercise, the outcomes comes major from cross-sectional studies, and they show the opposite. They say that athletes do not have higher bone mineral densities, they even have lower bone mineral densities, which is strange because they are exercising. So it has been estimated, for instance, in runners that 22 to 50% will suffer from osteopenia, and osteoporosis will range from 10 to 13%. So the literature, I mean, has come up with some reasons and some causes for this happens, and they focus that the reasons why these athletes are at risk of developing low bone mineral density is because they are exposed to some risk factors, like they are exercising more than they should be. That's one of the cause. They have low body weight, and that is because they are enrolled in sports. And because of that, they will get menstrual disturbances, and they have negative energy imbalance. So that will impair their bone formation. However, what happened is that other reports using the same populations as these ones has shown that even in the presence of these risk factors, there is not low bone mineral density. So low bone mineral density is not related to these risk factors, and athletes have increased bone health. So the question that comes up regarding athletes is that why does low bone mineral density happens in some healthy participants enrolled in sports activity and do not happen in others? I mean, they are exposed, let's say, at the same environment, so why ones will respond through a way and others will have a different response? So we feel that it's very important to answer this question because the prevalence, as I said, it's increasing. And moreover, sorry for this, that's a Macintosh, but the involvement in sports activity, particularly by females, have been increasing. So if we want to understand and use exercise as a measure to prevent osteoporosis, we need to understand what's happened to the ones that are exercising. And it's very important to answer this question. So the main purpose of all this project is to search for factors associated with low bone mineral density that might explain the different bone-anabolic responses seen in participants that undergo in organized physical activities. So in order to answer the question that I said, our physical exercise population was ballet dancers. And why ballet dancers could be a good model? Well, first, if we look at dancing, we can see that they have some activities like jumping and all the techniques that they require, all the techniques, dance techniques requires muscular strength. So this is known by literature to promote bone formation, to enhance bone formation. But on the other hand, you can see as well that in order to reach this level, they will have to start their exercise, their training at a young age. And since young ages, they are exposed to risk factors as low bone mineral density and intensive physical exercise, as the literature suggests. So they might be exposed to both an environment that will enhance bone formation, but in an environment as well that will damage your bone. So, moreover, if we look at dancing, we can see that it might affect differently the skeleton. For instance, you will have impacts on the lower body, so it's called the impact sites, because the exercise will have an impact in the lower body, but they do not use the upper body, so it's a non-impact site. So in order to understand the possible associations, it will be interesting to have this information at impact and non-impact sites. So what we did, we recruit dancers from two countries, Portugal and England, and we have professional dancers in each country, and we have as well dance students that were in a vocational dance school, having pre-professional training. And we have controls again for age and sex match controls in each country. So we measured bone mass, that was our first outcome, and we measured through DEXA. We measure all the outcomes that DEXA could give us, like bone mineral density, bone mineral content, and bone mineral apparent density. And we measured non-impact sites like the forearm and impact sites, that is the lumbar spine and femoral neck. We measured weight, height. In dance students, we measured as well maturation, and we use Turner sexual pubertal development and the biological maturing using the offset equation. And we record as well age at menarche in females. We measured energy expenditure, bone-specific loading as well, nutrition, hormones, and then we went for genetics. So I will not spend a lot of time explaining because I'm going through now. And we organize all the data that we collected in several studies, which I'm going through each, so I will not spend a lot of time now. So the first step that we did after organizing this project, it was trying to see how the bone health status of the dance students before getting the effects of physical exercise in dance. So what we did, we measured first-year dance students, so they were selected to go to the dance school, but they were not exercising. So we measured them before going to physical exercise. And we measured bone mass, we took blood, nutrition, anthropometry, and maturation as well. And we did the same in controls. Our results, and we compare by comparing dance students and controls, so at baseline, without effects of physical exercise, we saw that weight and height was significantly lower in our dance students. Dance students were doing some exercise before going to the dance school, around 2 hours per week of recreational dance, not professional, and controls were not exercising because it was a criteria. Calcium intake was significantly higher in our dancers compared with controls, and we can see here that according to Turner stage maturation, the majority of our dance students of the same age as controls, they were at Turner stage 1 and 2, while the controls were at more advanced Turner stages for the same age. So we measured bone mass, and we can see that bone mineral content, bone mineral density, and bone mineral apparent density, it's significantly lower in our dance students. We adjust these values for the Turner stage, weight, body weight, and calcium intakes, and after the adjustment, dance students continue to display significantly lower values at the femoral neck, at the forearm and at the femoral neck, while we cannot see any differences at the lumbar spine. So we concluded that before going to exercising, they already had low bone mineral density compared with controls. So, and the reason of this difference might rely in maturation because they are late maturers. That's why probably their bone have less mineralization. So the question that comes after is that, okay, we measured before going to the exercise, what happens while they start exercising, what happens to their bone mass throughout time. So our next step was to model bone mineral accruals throughout time. And what we did, the same students that were accessed before, we recruited additional dense students with at least 1 year of experience so that they can have benefits of the training. And on the second year, I mean the first year follow-up, we follow these dancers, we follow this one, we recruited more, and then they were measured at the same variables. And these groups again on a 2-year follow-up. So we followed for 3 years our sample. This is the general characteristics, I mean baseline, first year follow-up and second year follow-up. Weight was always significant lower in dense students, but the height difference was only significant at baseline. The age at menarche was around 1 year later in our dense students, and the bone mass, the bone mineral content values were lower in our dense students. So if we look at the graphs, we can see here that ballet dancers have lower bone mineral content throughout their age as they grow up compared with controls. This is for femoral neck. The same happens at lumbar spine, and at the forearm the differences are even higher. So we do not have a significant interaction here, which means that at baseline they have a difference, and that difference continues throughout the time. So dense students never go over controls. They always keep low values. So we're trying to find some predictors that could explain the bone mineral content gains and the differences between groups. And we first searched for traditional osteoporosis risk factors like weight, height and age at menarche, and they were playing a significant role at the femoral neck. So when we control for these covariates, I mean, the group difference does not exist anymore. However, there still is a group difference at the lumbar spine and forearm. So, and the dancers still continue to display low bone mineral content. So, which means that that the reason body weight could explain some of these differences, but it's not the only significant predictor that might explain these differences. So the question is, what might explain these differences between groups? So the process of bone remodeling that I said before, it's regulated by local factors and systemic hormones. So, and if you remember, as I said before, I mean, during growth there is an increase in sex steroids, and that stimulates the release of growth hormone from the hypothalamus, and then they go to the liver, and the liver produces EGF-1 that goes to bone. And EGF-1 is very important because it regulates osteoblast activity and reduces osteoblast apoptosis as well. And study says that the lack of growth hormone production usually is associated with low bone mineral density values. So if you remember, sorry, before that, so growth hormone and EGF-1, they decreases with age, but they are particularly increased during the growing years. So it's very important to have normal growth, in order to the levels of these hormones could be optimal, so that the bone can use these hormones. But if you remember, our dense students have delayed maturation. So our hypothesis, we're trying to think, okay, these hormones might be predictors of bone mineral accrual changes. So our aim was to investigate if circulating levels of estrogens, growth hormone and EGF-1 will be a significant predictor of bone mineral content changes. So this is our baseline general characteristics. You can see here there is not a significant difference in terms of estrogens. But there is a significant difference in terms of growth hormone at the first year follow-up. Dancers have low values, and there is a significant difference as well in EGF-1, and dancers have higher levels. So when we look at our model to find our predictors, when we control for the known factors that were playing significant role, like weight, height, and age and monarchy, there is not a significant difference in forearm, the femoral neck, but there is at lumbar spine and forearm. We found out that growth hormone was a significant predictor of changes in the femoral neck, and we found as well a group interaction. Means that this might explain the differences between groups at the femoral neck. However, we could not find group interaction in the other hormones. So again, we do not know what predicts bone mineral color changes at the lumbar spine and forearm. So we concluded, as I said, that growth hormone was associated with bone mineral content, but not at the lumbar spine, and at the arm EGF-1 plays a significant role, although however do not explain the group difference. So the question that comes up, it's still remains. I mean, what might explain the differences between groups? So a pretty obvious could be nutrition. Everybody knows that nutrition is very important, as I said, and particularly some concentrations of calcium, energy intake, protein could be playing a significant role. So we search for that, and our aim was search for that, and we can see that calcium intakes were higher in our dance students. Protein, however, were significantly lower, and there is not a significant difference in terms of energy intake throughout the 3 years. So when we go through our models, what happens is, again, calcium is playing a significant role in femoral neck, but again, it does not explain the differences at lumbar spine and forearm. So the question that came up was, is dance training a weight-bearing activity? I mean, is it an osteogenic activity? Because they are exercising, so it's supposed to have some benefits from that physical exercise. So, but is dance training relevant to bone? We tried to measure, because as I was saying, not all activities will enhance your bone. If you look at swimming is not an osteogenic activity. However, gymnastics is an osteogenic activity. Unfortunately, there's not information in the literature regarding dancing. We tried to do that in an indirect way. We could not do it directly. And here at the first year follow-up, we include the measurement of bone loading. And we did that again indirectly through a questionnaire. It's the BIPAC questionnaire, which is validated in the literature. It's used in several populations, and these participants are asked to record all regular physical activity, I mean current and past. And what this questionnaire does is, I mean, you enter the information on some equations, and it will give you a score for your the physical activity you've been taking, how much that goes through bone and counts as osteogenic activity. So of course, our dance students were with a higher score compared with control. So this is 67 against 37 and 20. I mean, this is current exercise, and 20 against 1.31. So, I mean, they are exposed to an osteogenic activity. So, the question is, okay, they are exposed to an osteogenic activity, but are they getting the benefits of that? So, in order to do that, if you look well at bone biology, there are several signaling pathways. Not only these ones, this was the most important. And if we look closely to these ones, we can see that these two are related to physical exercise. So when you exercise, you stimulate these two signaling pathways. And it's well known in the literature that some genes of these pathways are related to osteoporosis phenotypes. And particularly these ones here are well established in systematic reviews and genome-wide associations in several kinds of studies. So our aim was to investigate if there is association of some polymorphism, some genes polymorphisms of these pathways that might explain the low bone mineral density seen in our dancers. So here we recruited as well professional dancers, and all this group went through bloods, and we did a DNA analysis through blood cells. This is the signaling pathways, the estrogen receptor and the WNT pathway. This is the genes that we measure, our SNPs. This is their position, they are all in an intron position. And these three genes, actually, they were excluded for further analysis because they were significantly deviated from the Hardy-Weinberg equilibrium, so we do not measure it further, but all the others we moved forward with the analysis. We had to stratify our samples, so we have dancers with normal bone mineral density and low, and controls as well. So we have to stratify our sample. We couldn't do it in another way. What we found out was that when we compared dancers with normal density with dancers with low bone mineral density, we found a significant association in the A allele of the estrogen receptor 1. So the ones that have this allele have increased odds of developing low bone mineral density. We found another associations, but they weren't retained after a false discovery rate correction. However, when we compared controls with normal bone mineral density in dancers with low bone mineral density, we found out the same risk allele, but we also found out another risk allele at another gene, the LRP5. So we found here that these two alleles are increasing the odds of low bone mineral density at the lumbar spine, at the forearm as well, and at the femoral neck. So finally we found something at the lumbar spine and forearm. So we concluded that genetic variants at the WNT and ER signaling pathways are associated with increased odds of low bone mineral density in ballet dancers. Of course, we need further studies to confirm this, because especially our SNPs are in an intron and we couldn't find the biological significance of our SNPs. We don't know exactly what they are doing. So this might be the next step, understanding how the mechanisms that these two SNPs can be associated with our phenotypes. So the question, what might predict the bone changes between our groups? So it might be in genetic background, because we found the genetics was the only answer we found out regarding the phenotypes at lumbar spine and at the forearm. So the initial question, that is, why does low bone mineral density occur in some some exercising participants and not in others? I mean, our hypothesis is that the degree to which dancer's bone responds to mechanical stress from exercise training or dance training is associated with their genetic background. So, and what are the implications of these findings? So, if we look at the literature, again, as I said to you, and I bring this slide again to you, is that these are the reasons what the literature says that they have low bone mineral density. Out of this, I mean, we found that body weight was a significant predictor, not the only one. He could not explain everything, but we did find that significant association, but we didn't find like anything related to nutrition. But we did found these two risk alleles. So, and again, very important, because these alleles were predicting at both impact and non-impact sites. So if you look at the guidelines of the American College of Sports Medicine, they will scan particular women if they have low body weight, negative energy balance, and if they are enrolled in intensive physical exercise. And they will say, okay, if you have this, you are referred for bone densitometry, and if you have low bone mineral density, means that you have secondary osteoporosis. So what they do in these cases, they go for hormone therapy, and they go for nutrition supplementation, and they sometimes prescribe extra physical exercise. Of course, they say to increase your nutrition intake, but they do this. And surprisingly is that there is no significant effects. If you look at our population, which is dancers, there is some studies doing hormone therapy, and they couldn't find any differences after the hormone therapy. So if we follow our our results. I mean, this is something that we are still working on it. I'm still trying to understand what I have here. But if we look at our 115 dance students and 36 professional dancers, we try to measure them according to the guidelines. So we measured body mass index, we measured fat mass, energy expenditure, and we calculate the energy balance, we measure nutrition intake, and we found out that out of this population, 25% of dancers will have no risk factors at all. They do not have any of this, but they have low bone mineral densities. And 2% of dancers have these risk factors, but have normal bone mineral density. So what happens is that we said that, okay, this might be association with low bone mineral density, but probably there might be some associations, and these pathways probably need to be further investigated. Because if our results are showed in other studies, it means that we are in the presence of idiopathic osteoporosis and not secondary to osteoporosis, which means that in this case you don't go for hormone therapy or nutrition supplementation and not extra physical exercise. So of course further research is required to confirm our results, and of course we have as well to go through other exercising populations because this is only an example. So thank you for your attention.